Thank you. Regina, can you tell me if you agree or disagree to the statement that I just put in? Okay, cool. All right, we'll begin then. I'm going to share screens with you all. You should be able to see my screen in just a sec. Okay, can y'all see that? All right. So let's start by defining these different leukocytes here. I'll give you all about 10 minutes to do so, and then I'll show you what I have, and we'll talk about them and make sure you understand what they are. Y'all all understand what you're doing right now, correct? Regina, if you're talking, I can't hear it. I see that there's a microphone there. Yeah, just doing the definitions. That's all you're doing right now. Sorry, I don't know. It says there shows a microphone thing next to your name. I just don't see. I don't hear anything if you're talking. You have to click if you want to talk. There's a talk tab in the top left-hand side underneath that big dark box, and you can talk there. You have to click that to talk, I mean. Okay, so I will post, or I'll put on there what I got for definitions. Is that what y'all got for your answers here? if you see anything that's different or if you have a question about it, just ask me. Okay. Well, then you should be fine. Trying to find where they have the split chart of how everything made. Hold on. On page 434, figure 14.7, I would take a look at that. Just it helps you to understand how each one of those things becomes, like how basophils come from the myoblast, and basically it all starts from that stem cell, and then it just goes from there. So it's a good way to understand these leukocytes. I would definitely take a look at this figure here. We'll do something. Okay, can everybody see that? I think I'll make them a little bigger. There. Oops. Okay, this is dumb. All right, so can you? This is the first, second, and third levels of immunity, or immune defenses, I mean. Can you guys tell me? We'll start on the first. Can you tell me what goes in the first there, or out of this word bank right here, which goes in the first line of defense? Innate, right? Do you know what they mean by innate? Innate means that you're born with it. It's not something that's you acquire. You have it. So just remember, like, everybody's born with skin. Everybody's born with they should be born with skin. They should be born with your mucous membranes being able to form tears and all of that. Physical barrier, yes. Fever does not belong in the on the first level. What else? There's a lot there. Coughing, sneezing. I just did that. Oops. Anybody see anything else, or we'll just move on to the second layer, or second line of defense? Okay, so what about the second level, or second line? I keep on wanting to say level. Mostly non-specific. Inflammation. Oops. Yes, it is fever. You got it right. Interferon. Somewhat acquired, yes. That's why. Or no, it's not somewhat acquired. It's somewhat specific, but mostly non. But it's innate, because everybody can have everybody's born with the ability to have a fever. Everybody's born with the ability to have inflammation and all of that. Interferon, phagocytosis, right? And you said innate? Alright, so the third line. Not all of these go in the third line here. Which ones do? Specific. Acquired, right? I'm going to add another one too. IgG, what is that? It's, or I'll just tell you. So it's what you build up after you're exposed to something like a cold. Your body will get the infection, but it'll start to form antibodies that'll fight that infection if it comes back again. Yeah. So it's part of your antibodies So this would for sure be in the third because it's specific, so you don't even have to think about it too much. If it's specific, it goes in the third. Now, with that in mind, what about the natural killer cells? What do you know about those? Are they specific or non-specific? Yes, natural killer cells. They are innate. So what do you think they are? Second, right. And they're non-specific, so they belong in the second. That's correct. What about skin, tears, low pH, lungs? That's what it should be. Yeah, first. Tears as well, yeah. That's part of your physical barriers, the first line there. So before we move on, I want to go over, actually, I guess we'll do it after the chart, I guess. Okay, here's the antibodies chart. If y'all could fill this in on your own sheet of paper. Yeah, low pH that's actually on your low pH was on your first one here. Yeah, dang it, I already closed it. Let me open it up and see if I can find it. There. No, yeah, dang it. Sorry, I closed out of it way too fast. Which one, what did you need? Did you just need to write it down, or that list? Okay. Let me assemble it real quick. Okay. Well, if you can find it, then that's fine. Okay. Yeah. This is all out of Chapter 14. If you take a look at Table 14.1, it will pretty much tell you everything you need to know. So, going to Chapter 15, this chart right here, if y'all could fill that in, I'll give you about 10 minutes or so to fill this in, and then I'll show you what I have, and then we'll talk about what it means and what you definitely need to know for your test. Okay? Yeah, there's a review this Saturday and Sunday. And I think if you're in Cooper's class, Cooper is actually having a review on Saturday afternoon. Yeah, afternoon, I think. Oops. Yeah, it's table 15.2 is where it's at. Okay, cool. Yeah, it's been 10 minutes. So, I'll show y'all what I got. There we go. So the key here is to look at, for IgG, definitely know about this one right here that I'm highlighting. Very important one to understand. It has, or know that it makes up the largest percentage of your total antibodies in serum. Know that it binds to your phagocytes. It's a long-term immunity, and its life in the serum is 23 days. IgA, its biological function is it's a secretory antibody on a mucous membrane, so that would be like in your nose, anywhere where you have like a membrane. Let's say your snot in your nose would be a good place for that. Know that IgA binds to epithelial cells. because where would your mucous membranes be? That's in your epithelial cells. That's your skin cells. IgM, no, you don't need to know the shapes of all of these, but I would know that IgM is a pentamer, meaning that it has 5 antibody binding sites on it. It's kind of shaped like that star if you're looking at page 470. Know that it's the first response to an antigen, and we'll talk about what that means in a second. So know that there's only 6% of it in your total antibody, or percentage of your serum is that. IgD. IgD binds to is a receptor on your B cells, and it's responsible for antigen recognition. And then your IgE, no, that's your antibody for allergies and worm infections. So IgE is associated with your allergies. Just know that IgG, if you're thinking about like what do I need to know as far as like numbers, definitely know that the percentage is the highest in IgG. IgE is the lowest. Memory? Yeah, memory. And your long-term memory is IgG. So I would just know that for sure, IgG, and then know some about each one of these, because she's not going to ask you to regurgitate this chart, but I would definitely be very familiar with the chart. Do you have any questions about the chart? We'll wait until Regina says something before I move on. Oh, okay, good. She's having it after SIT Saturday. I don't know where. I heard that she's going to send out an email, but yeah, I think she's going to send one out tomorrow about it. No problem. Okay, take a look at the graphs on page 471, figure 15.13. So, can somebody tell me what's going on in these two graphs here? Knowing what you know about IgM and IgG. You're confused. Yeah, that's kind of confusing just by looking at it. See if either one of them know. Yeah, y'all are you're getting close to what's going on there. You're pretty close. So on that first graph on the left, it's saying the first exposure to antigen, and your primary response, remember, is for IgM is your first response to antigen. So you know that you're going to have a large amount of IgM produced when that first exposure to antigen has happened. You're going to have the lag period, and that's what that 10 to 12 days lag period is. And then you're going to have an increase in IgM, and then after that you're going to have IgG. So that's your first response. Then on your second response, since you've already met this antigen before, you're still going to have a little bit of IgM, but your response, your memory antibody, your IgG, is going to have a huge growth, and that's what's going to take care of your antigen the second time. That's why typically you don't get sick from the same thing twice, because you have a memory. Oh, well, maybe. Do you see where you're on now? 471. Okay, cool. Does that make sense to you? Or I can go over it again if you want to. Okay, yeah. Just know that your latent period, your IgM is going to build up, and then your IgG is going to come up. The second time, your IgG is going to be a huge response, and your IgM is going to be little. Okay, yeah. Does that make sense? I went a little fast, but hopefully. Because your IgG is your memory, your IgM is your first response. So, and again, this is why when you get exposed to the same pathogen or antigen once, you're going to typically not be affected by it twice. Yeah, it's not going to be there all the time. Like your antibodies for, I don't know, whatever pathogen polio, since everybody's had the polio vaccine, or I don't even, yeah. It has a lifespan. You don't have antibodies going around in your body all the time for everything. You just have a few, and whenever they sense that coming in, then they build up a huge response to it. Does that make sense? Yeah. Off topic, but if y'all have time, I know you have to study for your exam, but if any of y'all have HBO or have access to HBO, I think HBO, you can get it. I think you can actually get this documentary on YouTube. But Vice, I don't know if y'all watch Vice, but there's a show on HBO that came out that's basically talking about how they're engineering viruses to attack cancer. And it's kind of on this immunology subject, and yeah, they're using measles, and they actually used HIV. If you watch the whole thing, they were using HIV to basically cure cancer. And I mean, they're probably far away from this. I guess they're not too far away. They said maybe next year they're going to be actually coming out. But it's kind of interesting. Yeah, they're using basically viruses to attack tumors and cancer in the body. And now that you've kind of taken somewhat of an immunology brief overview of it, you'll understand it when they're talking about it. And it's pretty cool. Anyways, do y'all have any questions that you'd like me to, like anything over any of the subjects you've covered for this exam? Anything about the exam, anything like that? What charts do you need to focus on? I would definitely know or be very familiar with 15.2. I would know the general structure of a antigen binding site, like on 15.1. Figure 15, yeah, figure 15.1 has a good overview on the antigen and the antibody and everything like that. Kind of shows you what's happening. I would definitely go back over her, or what is Hansen's? I don't know how much she adds from the first test. It's not going to be a whole lot. It's going to be stuff, if she mentioned it all, that, did she mention that what was the most missed question? Because if she did that, then yes, that will probably be on the test. But it's probably not going to be a whole lot from the first test. She's going to wait until the final exam to really bring back the first test. But she will bring back some, just not a whole lot. For Chapter 9, though, for Cooper, definitely know that she gave you that DNA problem, the RNA, or the replication, transcription, and translation. Know how to do that, because that's probably going to be on your test. Know the different microbial controls. Know the difference between neurotoxin. Hold on, let me pull this up so you know that. Can you do this right here? I don't think I did. Can y'all not hear me? Oh, you have your mic thing on. If you go where to talk, click on the talk thing. I think your mic might be on, and that's why you might not be able to hear me. Oh, she probably can hear me. Oh, you can hear me now? There's some videos on the book website that can show you the different enzymes and everything. I would for sure know how to do, if you know how to replicate it, then you should be good. And then I would take a look at your quiz that she gave you and basically answer the question that she gave you on that quiz. The quiz about the replication, like describing it. Go online, go to your book, and there's a video on that. And I would look at that, and then I would also do her question she gave in class. This right here that I highlighted, she's going to ask you a question about this too on the test. That's why I'm giving it to you. People confuse these up a lot. What about IgG? Oh, can you not see my screen? Okay. Hold on. I don't know why it's not showing. Let's see. I don't know. Can y'all see it now? No? What do you see? Well, it's not telling me that y'all can see my screen. Resume. So you can see my screen here, the endotoxin, neurotoxin, yeah. Can you use this in a sentence, because this is for sure going to be helpful for Cooper's test, and I'm sure Hansen is going to ask you a question with these words in it too. So I'll just tell you what these are, or I'll give you a brief definition of each of these, and I'll see if you can use them in a sentence. No, I mean she might give it to you. I feel like she gave you a short answer question similar to this last year. That's why I'm giving you all this. So endotoxin. Okay. So there's two different types of toxins she talked about, and this was like maybe 1 or 2 lectures ago. And she said endotoxins are released once the cell dies. The toxin's located inside the cell wall, I believe. And when it dies, it breaks apart and that toxin is released, and the toxin just goes and attacks or affects whatever. It doesn't attack anything specific, it just affects anything that it touches. It's not attacking anything specific though. Exotoxins are secreted by a pathogen to attack specific tissues. Like if it's a toxin that affects the nervous system, it's going to be a neurotoxin. If it attacks the GI tract, it's going to be an enterotoxin. Does that make sense? So if you were going to use this in a sentence, you would say endotoxins are released once a pathogen dies. Once it breaks apart. exotoxins like neurotoxins and enterotoxins attack specific tissues in the body and it's secreted by a pathogen and it attacks specific tissues in a body. Does that make sense? I went really fast, but yes, entero and neuro is with exotoxin. Endotoxin, it's by itself because endotoxins don't attack anything, specific. Enterotoxin attacks the GI tract, so it's an exotoxin because it attacks something specific. Neurotoxin attacks the nervous system, so it's an exotoxin because it attacks something specific. Endotoxins do not attack anything specific, they attack whatever they're closest to. Yeah. And when I say attack, I guess that's just like by attacking, I mean the toxin goes to that area. It's just easier for me to say attack because you know that's what it does something bad to that area. All right. So do y'all have any other questions about what we went over or anything? I know it's not extremely clear, but if you have any questions while you're studying, you can always email me. I would definitely try to go to one of the study sessions. We have 2 that are run by us, and then there's one by Nanny. So if you can make it to one of those, that would be a good idea. All right, good. I'm glad. I am doing the Sunday one. Yeah. I'm not sure who's doing the Saturday one. Yes, it's 4 to 6. All right. And yeah, Saturday is from 10 to 12. That's correct. So if y'all don't have any other questions, I guess we can end this. Y'all can leave. Y'all have a good night and make sure you're studying and preparing for the test. No problem. Yes, good luck to all of you.